So hello everyone and welcome to GW4 AMR 2020. So this year's symposium is titled Multidisciplinary Approaches to AMR, Bench to Bedside and Beyond. My name is Hannah Bustrom and I have had the honour of being the chair of the organising committee. So I'd just like to give an introduction to the live sessions of this event. It's so wonderful to see so many people have been able to join us, and I know you're from so many places across the world. We have a really, we're quite impressed, quite happy with our jam-packed lineup of both invited speakers and submitted abstracts, providing short talks, flash talks and posters. And really the idea behind our event is to communicate and also collaborate on investigating and combating AMR from all the angles possible. So our original plan for this event, when we first started working as a team, was that it would be an in-person meeting hosted at or near one of the GW4 universities. So that's Bath, Bristol, Cardiff and Exeter, all based in the UK. However, obviously, a global pandemic gave us a reason to revise our plans. So this has been a very steep learning curve for us to be able to transfer our knowledge and sort of skills in putting on any kind of live event, which few of us had done already, to actually sort of changing it to a completely online event. So I hope you'll forgive us if we make any weird mistakes. We have done our very best here, and we're quite pleased with what we've come up with. So as you know, we have 6 live sessions happening over the next 3 days. So the session now, and then we have a short break, and another later this afternoon, 2 tomorrow and 2 the following day. And the programme is fully populated and available on our website. Also on our website is an attendees area, which is dedicated to everyone who has registered for the meeting. And there you can find galleries of the posters, recordings of the flash talks, the images of research gallery, and also we will post their recordings of the live sessions. Please bear in mind that some speakers, due to intellectual property or other data proprietary concerns, will not be having their talks recorded, so do join the live sessions, but if you do miss anything, it'll be there for you to replay or catch up with later. So as we all know, and probably because we're all here, AMR is a huge global problem, and for quite some time really, but definitely in recent years, it has become apparent that in order to tackle antimicrobial resistance, in order to control it and also reduce it, we need to go for a One Health approach. So we need to act from many different angles at once, within human health care, veterinary care, agricultural use, and all other all the other angles you can think of. You might also know that it was Antibiotic Awareness Week last week, so hopefully you're already feeling very nicely aware of antibiotics, and now we can have a look at the problems that come with the use of them and how we can hopefully help control and resolve these in future. So we hope that our meeting will be both informative through all of the talks that we are sharing and the posters and other information, but we also hope that it can be collaborative. So one of our key aims in transferring this symposium to being online was to ensure that we could still deliver an element of networking and collaboration and talk between attendees, because we didn't really want this to just be isolated webinars. So if you go to our website and you can join in on the forums and either continue discussions from these live sessions, anything that's raised during the panel Q&As, and also start your own topics. And we really hope that this will help networking, just sharing of ideas, helping one another if we need it. We realise as well that due to the pandemic situation, we're quite isolated, even perhaps from our own research groups and colleagues. So we thought this would be hopefully a nice way to, well, make new research friends and hopefully find ways of working together, or at least understanding one another and the different angles that we're all coming from. So I would like to say a huge thank you to the whole of the organising committee this year. You have been a wonderful team. I have loved working with every single one of you, and I think we have worked rather well together, and I'm really proud of what we have achieved between us. Everyone has gone above and beyond and really put in a lot of work into delivering this. So thank you everyone. Please everyone go and have a look at the About Us page on our website, so you can find us in the forums and congratulate all of my colleagues in how well they've done on this. That said, if you do have any issues with website access, with these meetings, or any problems at all, please do drop us an email. We will be manning our mailbox throughout, and we'll try to get back to you as soon as possible. Obviously it depends how many people are messaging us, but yeah, we are human and we are here to help. Before we get going, our new interim director of GW4 would like to say a few words to open the meeting. So I'd like to introduce Jane, and thank you for taking time out of your day and coming along to open this for us. Thank you everybody.
Thank you very much, Hannah. I'll just put up some slides. Hopefully you can all see that. Right, so hi everyone, and absolutely brilliant to be here today. Welcome to the symposium. I'm the interim GW4 director, and I just want to say a few words on GW4 Alliance and AMR. We were established in 2013, and we bring together four of the most research-intensive and innovative institutions within the UK. So we're the universities of Bath, Bristol, Cardiff and Exeter. And collaboration very much sits at the heart of what we do, and basically is what makes us greater than the sum of our parts. And the idea is that we aim to drive innovation and economic growth across our region and very much beyond. We have over 8,000 academic staff and over 23,000 postgraduate students. And it gives us a great opportunity to really harness a combined research income of greater than £370 million. There are lots of things unique about us, but one thing that's very unique is that we're the only research alliance that cuts across England and a devolved administration. So which means that we have two governments with a variation in policy, but also two health systems. The GW4 Alliance has invested over £2.9 million in 93 collaborative research communities, working with over 200 companies. Now this has generated over £46 million in research income, which means for every pound that GW4 spends on collaborative research, we capture over £15 in external research awards. And most importantly as well, together we're contributing to knowledge transfer and global impact. And this is through influence in national policy, developing world-class facilities, and also providing the foundations for major research programs. And now we're very much initiating research at scale, really, recognizing our combined research strengths. And some of the examples of the areas we're focusing on are AMR, climate, and net zero. You know, Hannah put this really well earlier. AMR is very much a priority for us, and Hannah gave a great summary earlier as to the importance of tackling AMR. You know, we know it's an increasingly serious threat to global health. We know over 700,000 people die every year due to drug-resistant infections. And we know it's one of the greatest emerging threats to human health. The scale of the socioeconomic threat of AMR is costly and wide-ranging. And the economic damage of uncontrolled AMR will be further compounded as a result of the current COVID-19 pandemic. The impact on food production, trade, livelihoods, and increased poverty and inequality are all a serious issue. Unimpeded development of AMR will have a catastrophic impact on global health, food sustainability and security, well-being, environmental well-being, and also socioeconomic development, a very critical area for all of us to be involved in. Now, we have several approaches when it comes to AMR. We've recently set up the GW4 AMR Alliance, which aims to tackle this global challenge with an interdisciplinary One Health approach, as Hannah said earlier, and unparalleled expertise. And members of that group are also part of the symposium today. Part of the mission of the GW4 AMR Alliance is to cultivate a collaborative interdisciplinary research and training environment in which the next generation of AMR researchers will flourish. So very much speaks to the heart of this symposium today. We also have the GW4 ECR Symposia scheme, which this is part of, this particular symposium is part of. It's a new scheme and it aims to create a space where PGRs and ECR, so early career researchers, can showcase their research, gain experience in managing the delivery of an online symposium, broaden their networks and form new collaborative cross-cohort interdisciplinary research communities. So this year, the focus is on strategic priority areas of AMR, as we have today, but also on climate. We've got an event starting tomorrow. We also have the GW4 Crucible, which brings together 30 competitively selected future research leaders to explore how they can enhance their career by working with researchers in other disciplines and how their research can have greater impact. This year, the theme was interdisciplinary approaches to AMR, and 5 seed-funded collaborative projects emerged from the workshops. So our institutions are home to world-leading research facilities and complementary expertise in AMR. Our proven academic excellence in AMR research across disciplines is also demonstrated by a whole portfolio of AMR relevant research funding, which is well in excess of 40 million. I give some examples there in terms of the areas that each of our universities focus on, but very much complementary expertise across the field of AMR. I thought I'd also mention that, you know, we host over 30 externally funded doctoral training entities and partnerships, so doctoral training centres and partnerships. These are very highly collaborative, involving a range of academic and non-academic partners from national research organisations, industry, business, government and the third sector partners. We're very much building a highly skilled workforce, and PGRs and early career researchers are the foundation of our vibrant research environment. So our alliance very much aims to learn from each other, and we work collaboratively to add value and continuously develop and update the support we provide to our communities. And just to restate this, really, we are very much committed to supporting our researchers across all institutions and all stages of their career development. There are many benefits to being part of GW4, which I'm hoping a lot within the audience have experienced to date. But there's access to expertise, skills, facilities, training and development opportunities, seed and generative funding, collaborative spaces and resources. And this is across all 4 universities and doctoral colleges. So very much a lot for our community, from early career PhD students, early career researchers, all the way across the people pipeline can gain from across universities. And before I finish, I just wanted to say a huge thank you to the organising committee and everyone who's helped organise this event. Please do make the most of the event, engage with the theme discussions, check out the online poster session and research gallery. There are some fantastic speakers lined up over the next few days, and I know you're all in for a real treat. I look forward to seeing new collaborations and ideas emerge from this symposia, and look forward to meeting some of you over the coming weeks and months. Thank you very much.
That's brilliant. Thanks very much, Jane. I'm just going to share my screen. So my name is Becky Weiser. I'm a postdoc in Cardiff University, and I'm going to be chairing our first session with my co-chairs Jamie and Charlie. Our session is One Health Approaches to AMR, and we've got some great speakers lined up. We have 2 keynotes who I'll introduce briefly before they each give a talk. Then we have 3 short talks that were selected from the submitted abstracts we have. We also have a panel discussion at the end, and then a short refreshment break before session 2. Just to give you a bit of housekeeping information about the session, if you would like to ask a question, please do so in the question box at the bottom of the webinar. You can ask questions at any point during the session. Myself and the co-chairs will collect the questions and then ask them to the panelists live at the end. If you'd like to ask a question to someone in particular, please put their name in the question so we know who to direct it to. Also, if you'd like to pose a topic for general discussion, then feel free to put that in the Q&A box as well. Everyone should be able to see the Q&A boxes and the questions as they are asked, and there's also the option of upvoting. So if you'd like to see a particular question asked, or if you had the same question in mind, then please press the thumbs up button and it will put those questions to the top, and we'll be able to ask them to the speakers. So I'm going to stop sharing my screen now and briefly introduce our first keynote speaker. So our first keynote speaker is Professor Matthew Avison from Bristol University. His research group looks into studying AMR in bacteria, and in particular looking at mechanisms of AMR in human pathogens, how AMR is spread and how it can also be controlled. We feel really privileged to have him as a speaker today. He's an amazing example of someone who's developed extensive interdisciplinary research networks and collaborations, and uses a One Health approach to combat AMR. And just to illustrate this further, he's involved in many different projects, both with microbiologists, but also chemists, structural biologists, physicists, primary care organisations, policymakers, and the animal health and farming sectors in this country and worldwide. So I'll now pass you over to Professor Avison, and we're really looking forward to your talk.
Hello, yeah. Thank you, Rebecca. That's very kind of you to say that. I'll try and live up to the introduction, I suppose. Right, let me just share my screen, see if I can manage to do this. And hopefully you can see that. And if I start the presentation. And can you see a single slide, or you're not seeing all my notes or anything?
Nope, that's all good.
Right, okay, excellent. That's always nice to see. So we're starting 10 minutes late, but I'll try and sort of keep reasonably to the brief of finishing in a reasonable amount of time. Yeah, so I'm involved in an awful lot of different projects, as Rebecca said, and this is one consortium that I lead, the OSTAR consortium. OSTAR standing for the One Health Selection and Transmission of Antibiotic Resistance Consortium. We've been going for sort of three or so years now, and this talk is really going to be about one part of that consortium, really, and that's considering the impact of farming on the local community. And the farming that we have mainly in the southwest in this context is dairy farming, so that's it. Now, I'm not a vet or a farmer in any way. Though as Tim Walsh will tell you, potentially I did used to keep pigs when I was a child, but this is really dairy farming anyway. So that means I build teams who do have expertise, and this is the team that was involved in the work that I'm going to talk about today. Of course, it was funded by the Cross Council AMR initiative under what they called Theme 3, AMR in the Real World. I'm not going to read all these names out. The three people really who did most of this work would be, I'll give myself a pointer while I'm waiting, would be Hannah Schubert, who's the vet who did all of the sample and data collection pretty much and kept 53 dairy farms happy for 2 years, which is not always the easiest task. Jackie Findlay, who did most of the genomics, particularly the whole genome sequencing, and Katie Morley, who did the most of the microbiology processing of all the samples. And then Ollie Mounsey did the phylogenetics that I'm going to talk about. Okay, let's start off by thinking about this issue. So what we're really thinking about is the idea of zoonotic transmission of resistant bacteria from a farming environment into humans and potentially impacting on human health. And of course, you know, we're all familiar with food poisoning and the fact that Campylobacter or O157H7 enteropathogenic E. coli are carried in the intestines of animals, and at slaughter those bacteria can cause contamination of meat, and if then the meat's not cooked properly, you can get a nasty diarrhea. Can be very serious. And of course, antibiotic resistance in those bacteria is likely to be driven by antibiotic use on farms. And of course, if that antibiotic resistance affects treatment of the serious diarrhea, then that can have an impact on human health. So we're kind of all familiar with that, and I know there's going to be a talk later perhaps about Campylobacter as well, so I won't dwell on this because actually we're not really interested in this project about the issue of food and that as a vehicle for transmitting resistant pathogens that have evolved on farms into the mouths of humans. We're more interested in the idea of the environment and environmental interactions, and whether or not there's any evidence of farming as a system causing an increase in resistance in the human population. And so, you know, dairy farms are a really good example of that, because of course most dairy farms don't produce a product that ultimately results in a foodstuff that's got lots of bacteria in it. You know, milk is pasteurized, or at least it should be if you're sensible, and therefore it's very unlikely that the bacteria that are living on dairy farms are likely to get into people via food. But of course, farms like dairy farms have got a wide range, and lots of people walk through them potentially on public footpaths, interacting with farm waste, you know, getting it on their boots and then potentially eating their sandwiches after taking their boots off. Or maybe they take their dogs for a walk and then their dog licks their face after licking them or licking something else. So, you know, this idea that there are other ways of getting resistant bacteria into you associated with farming, that's what this project's really about. And we're focusing on E. coli, as so many projects of this type do, because of course it's ubiquitous in the intestines of all mammals really, and lots of other animals. And so all of these species on the screen are going to carry very large amounts of E. coli. But E. coli is not just a curiosity, it's also a very important opportunistic pathogen, arguably the most important bacterial human pathogen in the UK in terms of serious disease and death. And of course, the association with antibiotic resistance is pronounced. You know, just sort of some figures, you know, a very large proportion of that 40,000 deaths a year from bloodstream infections might be due to E. coli. And of course, urinary tract infection is something we're going to talk about. Probably the most common bacterial infection in the UK, and 70% of those are caused by E. coli. So that's one of the reasons why we're interested in E. coli, because you've got this idea that if you encounter E. coli that's evolved to be antibiotic resistant on a farm maybe, and you become colonized by those E. coli, they can sit in your guts potentially for years and could be this kind of silent killer that could get you in the form of an opportunistic infection later on. And so that's potentially how this could impact human health. All right, so to study this, what we wanted to do is to look at a geographical kind of location where there is farming quite extensively going on, and also where we could get access to samples from humans as well. And so this is the OSTAR study site. These red blobs represent GP practices, and all of these GP practices will send urine samples for suspected urinary tract infection to the lab based at Southmead Hospital, just north of, just in the northern part of Bristol city, and those urine samples are processed to recover the bacteria and to determine antibiotic susceptibility. So we can get access to bacteria from all of these GP practices in a single lab, and that's what we've been able to do. But of course, within this range as well, there are also farms. There are also people living with their pet dogs, and I'm sure Jordan is going to talk about that later. And I'm not going to mention dogs very much, but this project will also look at things like domestic pets as well as farm animals. So this is our study site. That's Bristol, that's Bath. So it gives you a kind of a sense of scale and the amount of driving that was involved in going around this study site. So I just want to give you some kind of results. Basically, the first thing that we're interested in is, what are the drivers of antibiotic resistance on the dairy farms themselves? Of course, the whole point of knowing this is that you could potentially suggest some solution to the problem. You know, what could the farmers do differently that would potentially reduce the amount of antibiotic resistance? So even before we've established whether it is a threat to humans, you know, it's clearly a potential threat to the animals. So there's a benefit to reducing AMR on farms, not just because of the potential for zoonotic transfer. So the work I'm going to talk about today has all been published fairly recently. In this case, it's actually pre-printed, so I've given the preprint numbers for each one, and this paper is now in its kind of final stage of hopefully being accepted before Christmas. That'll be a nice Christmas present for us. Basically considering the risk factors for the presence of antibiotic resistance on these dairy farms. So I just want to give you some basic overview. First of all, we collected 4,500 samples. These are basically fecal samples from around various different animals on these dairy farms. It's a huge undertaking. We did this basically by going to each of 53 dairy farms every month for 2 years. So it's a big job. Of the 4,500 samples, some of them didn't grow any E. coli, so that was a bit of a disappointment. We threw them away. Of the ones that did, we basically looked to see whether we could find resistance to various different antimicrobials that are commonly used on dairy farms. So as you might imagine, amoxicillin resistance was reasonably prevalent, and tetracycline resistance was reasonably prevalent, and streptomycin resistance a little bit less prevalent. But you know, nobody was particularly surprised to find that these were quite common. They're common in humans. They're known to be common in animals in terms of E. coli resistance. What is less common, of course, is resistance to what we call the critically important antimicrobials, so the third generation cephalosporins and the fluoroquinolones, and indeed that's what we found. In fact, this would be less than you would find in your average person in this region. So I always say I'd rather be stood next to a dairy cow than a person on the tube, basically. But nonetheless, this wasn't particularly surprising. What was very interesting, however, is when we broke down the samples based on the animals that were associated with those samples, we found this very interesting observation that the prevalence of resistance was always higher in pre-weaned calves. So if the animals were young and pre-weaned, little cute calves are factories for antibiotic resistant bacteria. And as the animals got older, it didn't really matter how much older they were. If they were weaned, so sort of 2 or 3 months old, beyond that point, all the way up to 5-6 year old milking cows, the antibiotic resistance was lower, but very similar across all of those different age groups. So that was just the first thing to say that was kind of interesting, really. if you look at farm level, so that was individual sample level data. If you look at farm level data, then, you know, again, resistance was found to be very common across all of the farms. But remember, we were taking samples monthly over 2 years. It wasn't that we saw 100% of farms were positive for amoxicillin resistance every time we visited. It was just that at some point over the 2 years, 100% of them were positive. But I guess that's not that surprising. If you go more often, you ask the question more, you're more likely to get the answer occasionally. But what's actually quite strange, I found actually, was that quite a few farms, maybe 25% of farms, didn't have any third generation cephalosporin resistance in E. coli, even though we went all those many times and collected all those hundreds of different samples. 25% of farms didn't have it. So there's clearly some variety, and particularly that variability in the critically important antimicrobials, which is kind of nice because they're critically important for a reason. The other thing that we monitored at the same time as antibiotic resistance over this 2 year period was their usage of antimicrobials. And there's been a general trend to reduce the usage of antimicrobials in all farming in the UK, and they've been incredibly successful in doing that. In dairy farming, similarly, there's been a reduction. The blue is just a general usage of antimicrobial in defined daily doses per animal. It's the gradual reduction. And this is the critically important antimicrobials at the bottom in red. And you can see that by October 2018, when we kind of finished, it was zero. The farms were not using any critically important antimicrobials. And actually, the reason for that is that Red Tractor, which is an organisation that you've probably seen if you buy meat and it's stamped on the packet, is a kind of a system for overseeing what goes on farms. And one of the things they've done is basically effectively banned the use of critically important antimicrobials on these farms, unless there's absolutely nothing else that they can use to treat a particular diagnosed infection in a particular animal. So our farms were gradually moving in that direction, so they weren't that shocked when all of a sudden they were told, actually, if you want to be a Red Tractor farm, and 90-odd% of all farms are, then you have to stop using all of these critically important antimicrobials. So it shows that this sort of consumer and driven policy kind of works. Anyway, antimicrobial usage reduced, but it's still quite variable. So these are all of our 53 farms, and the amount of usage, you can see some of them are very low and some of them are very high. So we've got that nice variation that we can see. And so we could factor that in to try and understand why some farms may have more resistance than others. So that's what we did. We basically did a risk factor analysis using farm management questionnaire data. We asked the farmers 600 and something different questions about the way they manage their animals and the way they manage their farm in general. We looked at the antimicrobial usage data based on purchase from vets. We looked at the sample characteristics, so where did the samples come from on the farm, and we looked at weather, meteorological factors as well. This slide is very busy, but it summarizes basically a Bayesian regularization approach to risk factor analysis. I won't go into the details unless you push me, but basically it's a complicated way of factoring in all of those different things that are going on all of these farms, and with resistance as an outcome, trying to establish what were the primary drivers, the primary variables that can predict resistance as an outcome. And this is all small text and you don't have to read it, but basically the point is resistance to all of the antimicrobials that we looked at. The biggest, most significant driver that we identified was that the samples were collected from calves. Well, we already showed that really in a very simplistic analysis, but then next that it was all about temperature. So the average monthly temperature that we collected the samples. So samples collected in winter are much less likely to have antibiotic resistance. And this wasn't because they had fewer E. coli, because all the E. coli were sort of shivering and dying. That was factored in. It was the proportion of E. coli that were resistant was lower at lower temperatures. And the other thing that was found for most, but not all of the antimicrobials, was if we took a sample from pasture lands out on the public footpaths, then actually there was less resistance generally per unit of E. coli than there was if we collected it from within the housing, within the farm. So I'm not going to read all these are the same for all of these. Higher temperature, higher resistance. Younger animals, higher resistance. Out on the pasture land, lower resistance. Okay, and this leads us to kind of this really interesting kind of observation, which is if you collect samples on a farm, and you just go once, when you go makes a big difference as to what you get back. So for example, in February, we went and looked for fluoroquinolone resistance on 53 dairy farms. We collected, you know, several hundred samples, and we got 0 that were positive for resistance to fluoroquinolones in February, whereas 30% or 25% of the farms were positive for resistance in August. And we found pretty much the same thing for CTXM, third generation cephalosporin resistance, virtually 0 in February, 25% in this case in October. So, and the dots represent the average temperature. Those of you who know the UK know it's coldest in February, and that's when the resistance had disappeared. So when you go and collect samples will impact on the results that you get. So we can conclude a couple of things. One is that antimicrobial usage was a very weak signal. We did see a couple of very weak signals associated with very specific types of resistance that I won't go into due to time constraints. But generally speaking, the main thing that affected was where the sample was collected and when the sample was collected. Okay, so we would advise, if you're going to go and collect samples in the UK from farms or the environment or anything, that you need to make sure you always think about the site and that you're always going back to the same site, because different sites within the farms with different animals present will have different amounts of resistance. You need to think about going repeatedly during the year and not looking at farmer A in May and farmer B in February. You know, that's not going to work. You're going to get different results, and it's going to confound a lot of the analysis you're going to do potentially. Okay, so that's the kind of drivers part, you know, what's generating resistance. Now we just want to briefly talk about transmission of resistance. Is there any evidence that the resistance that's being brewed up on these dairy farms and then potentially being released into the environment, or people are encountering it as they walk across those public footpaths, is that actually going to cause a problem for those humans that live in that vicinity? And here we're going to use third generation cephalosporin resistance as the example. We've also done this for fluoroquinolone resistance, but we haven't finished analysing the data yet. So this is what we've got. And it starts off really with this published survey of third generation cephalosporin resistance in E. coli in our region in humans. So this is where we went to that lab at Southmead Hospital. We collected all the E. coli that were resistant to third generation cephalosporins that came from urinary tract infections in people in our region, and then we did a survey using whole genome sequencing to identify what sorts of resistant organisms are present, what types of E. coli and what types of resistance. And I won't bore you with the details. It's published and you can see it, but the point is we were able to characterize the baseline of what's circulating in the human population in this region, and then we could use that to compare it with the organisms that we found in our survey on the various dairy farms that we went and looked at. And so this is the first analysis. So this is looking at E. coli that produce mobile resistance. So this is CTXM beta-lactamase and AMP-C type beta-lactamases that are plasmid mediated. Actually, some of these are embedded in the chromosome, but essentially these are mobile resistance genes, which are the dominant reason for third generation cephalosporin resistance in E. coli. And so this paper, which has just been accepted in Applied and Environmental Microbiology, the ASM journal, shows what we did with mobile resistance. And this paper, which was published in Journal of Antimicrobial Chemotherapy, looks at AMP-C hyperproduction. So E. coli has a chromosomal beta-lactamase gene called AMP-C, and by mutation, that gene can be activated and be switched on, and then you get third-generation cephalosporin resistance as a result of that hyperproduction. So one's mobile and one's mutational, and we published these as sort of separate papers. Now, the overall analysis was that we looked at 1,200, just over 1,200 third-generation cephalosporin-resistant E. coli from those dairy farms. We used PCR to identify whether they have CTXM or whether they have a plasmid-mediated AMP-C beta-lactamase. As might be expected, CTXM was very common. That's true in humans. But actually what wasn't expected really was the AMP-C hyperproduction. So the mutational type of resistance was almost half of all of the third generation cephalosporin resistance in E. coli on farms. In humans, you would get about 85 or 90% would be mobile, and maybe 10 or 15% would be AMP-C hyperproduction. That's what we found in our survey of humans. So already we're seeing a sort of slightly different balance of resistance types on the farms compared to what we're seeing in the local people. But of course, this is just doing a few PCR. Well, I say a few, you know, it's a lot of PCRs, but it's doing some PCRs. Actually, really, what we want to do is do some whole genome sequencing to identify the organisms, and we couldn't do that on all 1,200, so we did a selection, 184 resistant third-generation cephalosporin-resistant E. coli that represented across all of the farms, and that tells you exactly what resistance gene it is, and it tells you the sequence type of the organism, and you can then do phylogenetics to look for sharing of those organisms. So the first thing that we observed was that CTXM32, which is not a particularly well talked about CTXM variant. It's a beta-lactamase variant, which is, you know, it's known about, but you look in the literature, there's a dozen papers about it. It's not commonly identified, and yet we found it in 79 of the 184 E. coli that we sequenced. What's more, it was found in 27 different farms. They were actually clustered together relatively closely geographically, and it was found in 25 farms, 27 sequence types of E. coli. So this is clearly moving around a lot, both from farm to farm, but also amongst the E. coli. And in fact, we identified that this is it's always the same plasmid. This is a single plasmid that's moved around from organism to organism across these farms, and we called it PMU32. Don't blame me for that. But basically, this PMU32 plasmid, it carries CTXM32, it carries resistance to streptomycin, and it carries resistance to other aminoglycosides and to tetracycline. So it's covering most of those antimicrobials that we looked for in terms of resistance on these farms, apart from the fluoroquinolones. This doesn't give resistance to fluoroquinolones. So it's a single plasmid that's moving amongst and between isolates and amongst and between the farms. And so the obvious thing to do was to see whether or not these isolates were similar. And not similar just to each other, but also similar to the humans. So this is a phylogenetic tree showing in blue the farm isolates, and in urine colour the urinary tract E. coli from humans in the same geographical region collected over the same time. And this was this shows you that the farm isolates are very, very similar to each other, lots of evidence of transmission, and the urinary isolates are very, very similar to each other, lots of evidence of transmission, but actually there was no evidence of E. coli that was the same in the humans and on the farms. Okay, there was one that was a little bit similar, but actually it was about 600 SNPs different, which is quite a long way in the grand scheme of things. There was some evidence of a few plasmids, 3 different plasmids that may be moving between human and farm isolates, but it's like 1 farm isolate and 2 humans, or 1 farm isolate and 6 humans. You know, these are not big numbers of plasmids, and these plasmids are known in the literature to be very promiscuous and very common. They're found on many different continents in many different animals, and so it's not surprising that these were the ones that came up. The PMU32 plasmid was not found in any humans. So even though it's found on all of those farms and it's been slewing around, it's not found in any humans. And then just finally, the same thing was really seen for the AMP-C hyperproducing E. coli. In this case, the humans are in green, and the human isolates and the farm isolates were quite different. Okay. So just to conclude on that, lots of evidence of farm-to-farm spread of resistant E. coli and their plasmids. Lots of evidence of human-to-human spread, but not really any evidence of recent acquisition of farm bacteria by people, at least not that have caused urinary tract infections, and a very small amount of evidence for plasmid sharing between people and the farms. Okay, and I'll just flash up those names and our funders, and thank you for your attention.
That was a great talk. Thanks very much, Matthew. I'll now briefly introduce our second speaker, who is Professor Tim Walsh. Professor Walsh has been studying AMR in bacteria for over 20 years. He's recently relocated from Cardiff University to the University of Oxford. He was PI of Barnard's, a Gates Foundation project on AMR, which looked at the burden of neonatal sepsis in low to middle income countries. He also leads several other projects examining the burden and spread of AMR in many different countries worldwide. He's a lead microbiologist to the Fleming Fund Expert Advisory Panel and an advisor to the World Health Organization, the Chinese Center for Disease Control and Prevention, and Médecins Sans Frontières. In recognition of his work, he was awarded an OBE for microbiology and international development in 2020. And I'd like to say thank you very much for joining us, Professor Walsh, and I will pass over to you.
Thank you. First of all, can everybody see my slides?
Yeah, we can see your slides.
Good, excellent, amazing. And just to a few caveats. First of all, is I've got roadworks going on outside my house, so if you hear some horrible noise, it's not my stomach. Secondly, is the fact that I've been asked. I thought it was a good idea to talk about AMR and COVID, but like most good ideas. At the time when the reality sort of has some credence near the time of giving the lecture, you suddenly realize that it was a particularly bad idea because there's an awful lot of things to do and a lot of kind of background reading to do. And I'd just like, the other caveat I would just like to say is it's a long time since I worked in a virology lab and worked as a virologist. That was in the late 1980s, early 1990s. And so whilst I know a little bit about epidemiology and I should know stuff about bacteria, I'm not a specialist in COVID itself. So anyway, in terms of AMR, and I was very pleased to hear Hannah and others talk about the GW4 and about the vision of it with respect to kind of a global outreach and about One Health and so on, is the fact that we've now, I think, come to realize, particularly over the last 5 or 6 years, largely thanks first of all to the Cameron government with Sally Davis and others. Regrettably, the UK momentum has somewhat diminished because of distractions with Brexit and so on, but nonetheless there was this visualization of the importance of AMR across the world. And you've seen these quotes before, and you had the UN Charter that was signed in September 2016 with 193 signatures on the paper. One of the biggest threats to human health, food security, which Matt has already spoken about, and really development today. And for those of you who don't know, there was the O'Neill report. In fact, Lord Jim O'Neill wrote several reports, of which one I'll be referring to during my talk. So the thing is, you know, why is it getting worse? And I think Matt's really given me a very nice introduction to that. Is the fact that we carry lots of bacteria as normal flora, And for those of you who are old enough, 20 years ago, when you were at meetings across the States and wherever it may be, nobody was interested in gram-negative resistance at all. It was all to do with gram-positive resistance. And then basically in the early noughties, mid-noughties, you then had, as Matt has already mentioned, ESBLs, particularly CTXM variety, and that took off like wildfire. It really did. Then you had the advent of really NDM, although KPC was around, but it was really a USA, Israeli non-entity, quite frankly, on the global scheme of things. Although there were sort of a mini epicenter in China, but it didn't actually have the global reach of NDM. And NDM was around, well, we discovered it in 2009, 2010, and the rest is history, as they say. And then we have, of course, MCR, which I'll mention briefly in my talk, which started off in China, rather like COVID, and spread very quickly around the world. And now we have TET-X genes that mediate resistance to tetracycline. And so all of these are in gram negatives now. And the sad thing is that we don't actually have any novel or good drugs for gram negative bacteria. And that's the tragedy of it. So whilst gram positives are covered, gram negatives are not. So in terms of the getting to grips with infectious diseases caused by the bacteria, is that bacteria are very complex, unlike viruses, which are not so complex, much smaller, much simpler organisms. And whilst there is genetic drift and shift within viruses, no doubt, such as flu, for example, is that the way in which they arrange their DNA and share their DNA, as Matt has already alluded to, is just simply not present within viruses. And so you have a range of vaccines then, I'll talk about them in a minute. And so essentially the key pathogens will continue to cause life-threatening infections that will go on to things like septic shock, etc., which is actually what my sister passed away with about this time last year. This is the WHO list of vaccines, and I'll just use my marker. Oh, look, there we are. And so these are the ones that are currently available, and these are the pipeline-like ones. And you can kind of look down here, and you can see, well, okay, good, Staph aureus may be covered, strep pneuma, which is a very obvious candidate or target for vaccine development. But the vast majority of these are actually viruses, not bacteria, and the ones that are bacteria are in fact gram-positive bacteria, not gram-negative bacteria. We only have one there, which is the enterotoxigenic E. coli. And so vaccines are not going to actually dig us out of a hole when it comes to AMR. For those of you who don't know, the WHO have a large program on vaccine development, particularly with protecting against neonatal sepsis, and we're helping them with respect to the design of the vaccines to the common or most common epitopes. In terms of the COVID vaccines, there are actually 55, I didn't know that until last night. There were actually 55 vaccines available, and these are the most common ones, and most of you already know about AstraZeneca's partnership with Oxford University, and you've got the Pfizer one, which is the US one, and so and there's one in China, for example, and the Russians have their own vaccine. But nonetheless, it looks like hopefully these vaccines will dig us out of the enormous hole that we managed to find ourselves in. So in terms of AMR, why will it get worse? Well, the fact is that it's all due to economic globalization, and there's a very clear overlap between COVID, or the spread of COVID-19, and AMR. It's the fact the world's become a very smaller place, and we travel a lot. And essentially the spread of COVID-19 out of Wuhan, Hubei province, was due to travelers going to and from China. There's also sort of I think a lack of global leadership, and that is also one of the reasons why I've joined WHO, is to try and help bring hopefully some new ideas and some energy into trying to think about how we can engage and pick up really on the UN agenda for reducing AMR. We also, as at the bottom there, I've already mentioned, we lack good pipeline antibiotics. So, you know, why is the burden global? And it's kind of interesting if you have a look at where COVID is at the moment, and roughly, depending on who you read, or what you read, who you read, and what sort of information you get, that the burden of COVID probably sits somewhere between 20 trillion and 40 trillion. But that's actually due to our response, not due to COVID itself, but due to the global response to COVID, in the fact of furlough and lockdown, and the fact that we are so fixated with preventing the spread of it that our economies are being hit and trade is being hit, etc., etc. The burden, estimated burden of AMR is actually 100 trillion, and that is from the report of Lord Jim O'Neill. Given what's actually happened with COVID, I think we can probably think that is an underestimation, not an overestimation. In terms of mortality, I mean, at the moment, I think we sit at about 1.45 million deaths, and AMR is expected to cost us roughly about 10 million deaths a year. I think that's also an underestimation, because we simply don't have the gravity or the rigor of the data from low middle income countries. The other thing that we have to think about, and this is also aligned with COVID-19 and how it spread, is how we actually feed our population, our global population, which will go up between an estimated 2.1 and 2.4 billion by the year 2050. And so the Earth is getting larger. We know where those economies are growing, mainly in South Asia and particularly Southeast Asia. And so they will, and the middle class is growing, and so therefore there is the demand on meat. So just again, going back to Matt's point about how we actually use antibiotics in agriculture is really, really important. And the vast majority of the meat trade, at least in that part of the world, is poultry as opposed to beef and pork. And the other problem that we have, which often is not, has been factored into this and overlooked, is the fact that we have global warming, regardless of its cause, but nonetheless we have global warming, and that will give more erratic weather patterns, which will spread AMR continuing around the world. These are just Worldometer data looking at the kind of the global incidence of new cases of COVID and deaths. And this is the lines, the solid lines are a 7-day moving average. And it's quite interesting that although we have far more cases here, we actually don't have too many more deaths. And a lot of these deaths that you'll see towards October and November have actually happened in South America, whereas the ones that are in March and April are essentially North American and mainly European. And that's simply because we're able to know a little bit more about COVID. We're not panicking as much, and we're able to manage patients a lot better than what we were, let's say, 6 months ago. These are just, this is a really busy slide, and I'm not going to spend any time on it at all, but these are just simply to illustrate this point that this is the NHS guidelines for managing COVID-19 patients. Now, back in March and April, we never had these guidelines. So in terms of sort of general management and respiratory support, we're a lot better equipped, and we know what to do. We know what's testing to be done. And if you think way back in March and April, the whole thing was just simply hitting the panic button. This also goes to the sort of antibiotics that we would give. So basically, COVID is an acute respiratory distress syndrome, and so we're able to recognize the symptoms more clearly. We're able to isolate the patients better, and we're able to manage the patients better. If you have a look at the antibiotics that are advocated for this, these are very simple antibiotics compared with the ones that were used back in March and April, where basically meropenem, aminoglycosides, you name it, it was given to patients who were presenting with these respiratory distress syndromes. And so we weren't actually denoting COVID from, say, flu or secondary bacterial infections. So there was a lot of panic going on. This is a study that I wanted to pull off and just simply show you. It's a very recent one, and this is just to illustrate the demographics of the patient population that are mostly affected by COVID. And if you have a look at in terms of the years, there is very much a very high risk in terms of years, as indeed there is body mass. It's very interesting actually that if you think about the group that could be most affected by this would be smokers, but in fact it's not, simply because the nicotine and the tar actually damage the very epithelial cells that actually COVID uses to bind. And so there seems to be a protection element with smoking. I'm not suggesting you all go out and lose weight and take up smoking. Although there was an interesting documentary I saw, a commentary, I should say, in a newspaper suggesting that if COVID had hit in the 50s and the 60s, where people were much slimmer and smoked more, then it wouldn't have had the clinical impact it has today. But there are other risk factors, not least about general mortality and things like immunodeficiency, etc. And this is just looking at the multivariant analysis with HR types. And as you can see, body mass plays a big part, again, as does age. That was very interesting that we had, as everybody knows, in the summer, we had this conversation about whether ethnic minorities were more at risk from COVID-19. And there was some interesting studies that came out of Queens in New York, which suggest, in fact, that that's not the case when you take into account multivariant analysis, such as overcrowding and obesity, etc., etc. And so that doesn't seem to particularly affect that particular group, that subpopulation. So this is an article, and I've already mentioned these figures before, and this is just the most recent one I could find, January the 1st, 2020, and there are various reports going around the world about how much this will actually cost, how much GDP, how many years it will take us to recover from this. And you've heard from the Chancellor that it's probably going to take about 3 years for the UK economy to recover from COVID-19, regardless of what may happen with Brexit. AMR will fall on the burden of low middle income countries. And we kind of know that for a very long time, largely because it's associated with poverty and also lack of sanitation, etc. And you've also got these other risk factors like the way in which antibiotics are used in humans and animals. So looking at MATS data, which is super clean, although they got an issue with, for example, CTXM32, which is interesting, that nonetheless is that the way in which we use antibiotics in these countries is considerably more responsible than what it is in low income countries. But nonetheless, low income countries will take the brunt of the hit. That's also because of the funding that they spend on, in terms of GDP, on their health systems, which is usually below 1%, as opposed to the UK, which is about 8.6, 8.7%. And in Scandinavia, it can be over 10%. So this is just an article that I thought I'd show you, and it's probably an article you haven't seen, one of the few you haven't seen, from Jim O'Neill. And it's interesting how he's suggesting that actually the way in which we use antimicrobials and the way in which we do environmental pollution actually drives antimicrobial resistance. And again, I was interested to hear Matt's talk with regard to the risk factors. This also, of course, comes down to how how COVID-19 started, and depending on your theories and your conspiracy theories, but the general feeling is it probably came from a bat or a market that sold bats in Wuhan in Hubei province, which is very typical of the meat markets in Hubei province. And the Asian population often is not eat all sorts of animals in all sorts of manners, and so therefore you have this very, very close contact. This kind of just looks at the spread of COVID-19 from the early stages across the globe, and so the epicenter clearly was in China, and then it spread into Europe, and also Iran got hit very, very quickly. and severely, as it does, still is. And then you can see that once it landed up in Europe and also the States, going from west to east, it then basically, you then have this explosion within Europe, and you also then have the explosion within the United States. The one thing that's very clear from the spread of COVID is that population density is the thing that unquestionably drives it. And this is just, looks at the severity, this is just a heat map of where we are. And it's very interesting because if you look at this heat map, it's almost entirely the opposite of what you see to AMR. So in antimicrobial resistance in terms of burden, you'd be looking at South Asia, Southeast Asia, still China, although China with COVID-19, although that turned out to be the start of the index of it, potentially anyway, China has managed to resolve this very quickly. And in fact, the epicenters, the two that you have are really Europe and also North America. And in fact, that's entirely the opposite of AMR. And so there are very clear differences with respect to that. And that just sort of is borne out a little bit by our aggregated data, which is, you know, a very crude way of looking at AMR. And you can see here actually that the epicenters are South Asia, Southeast Asia, and some parts of Africa where we actually can get the data from it. So in many ways, AMR is similar to COVID-19 in terms of the origin of where it spread and how it spread, and this connection that we have with animals. And this is borne out in terms of AMR, as this is just one example that we could give for many, is the fact that MCR, mobile colistin resistance, started off almost certainly in China way back in about 1987. And it spread from one organism into E. coli. We don't know what that organism was, probably Neisseria, spread into E. coli, and then the Chinese started to use colistin as a growth promoter about 10 years ago, and then it just shot up. When that happened, everybody started to look for MCR. And so this is, if you like, the spread of MCR within a 4-year period. So AMR doesn't quite have spread as quickly as COVID-19, or maybe have the clinical impact, but nonetheless, there are comparators for that. The good news is, which you won't see, of course, with COVID, is that when you start to reduce antibiotics, a bit like what Matt was suggesting, like, for instance, reduce colistin, you actually do see a reduction in colistin resistance around the farms, both chicken and also pigs, quite a substantial reduction. So that is the good news in terms of stewardship and the way we use antibiotics, particularly as growth promoters. Now, the thing that really is of note is that when we speak about antimicrobial resistance, we have this rather ludicrous phrase, which actually really quite annoys me, about AMR spreading into the community from the hospitals. And in fact, when you go back and have a look and think about these things in terms of resistance mechanisms, I can't think of one resistant mechanism that started in a hospital. They all started in the environment. They all started either with animals, etc. And in fact, this study, which was probably their best work they ever did, of Perel and Nordman, is where they definitively showed that you had the spread of CTXM15 here, where they looked at, put it together with an insertion element, which they called ECP1B, and they show that you can mobilize the gene from Cluvaria into E. coli. Beautiful piece of work, really elegant, really nice, and I think clearly demonstrates that in vitro that you can actually spread these genes from essentially environmental organisms into clinical bacteria like E. coli. This, of course, is not just with respect to resistance, but infectious diseases across the world. And so this is just an interesting article. It's quite a few years old now, but it's actually one of the most comprehensive ones. And what it looks at is the various viruses, for example, and the possible origins and the so-called new host. And the new host is an obvious new host, but you get a sort of adaptation that happens by the organism to take on its new host. And the interesting thing really is things like HIV. Now, HIV, really, it was very similar to simian virus. And when they started to look at polio vaccines in the 1950s, almost certainly the cases, the new cases of HIV from the Belgian Congo sort of simulate or could be transposable, if you like, on where they actually had the polio virus, done by Kaprowski's group back in between 1956 and 1958. And so it looks like, in the fact, that the way in which we may have produced the polio vaccine may have unwittingly released things like HIV. But nonetheless, what that suggests is that the way in which we worked with particularly chimpanzees in this case may have actually unwittingly released another type of infectious disease. So we maybe got rid of polio, but actually we started or produced something that arguably was as serious and as dangerous. And so, rather like AMR, we have this rather strange relationship with viruses or other infectious diseases and animals. And this is kind of borne out in this. So you've got bats, which we think we started with COVID-19, and bats was the origin of things like Ebola, went into other primates before making its way into humans. This is also borne out as well if you go back in time and look at the origins of H1N1, and you'll see that actually this really started over a century ago, in fact, where we started looking at the transfer from birds, and then you see the development of this all the way up to 2007, 2008, when you had, and up until about 6 years ago, when you had the latest outbreak of H1N1. And in fact, for those of you who don't know, the African swine flu was wiped out about 85–90% of the Chinese pig population. That only happened in about the last year and a half, which has kind of fallen off the radar because of clearly other things. So in terms of conclusions, I just like to say is that that COVID-19 there are similarities and there are differences. And the one thing I didn't really touch on in terms of the differences is the population. I mentioned the fact that the highest mortality with COVID-19 are primarily people over the age of 70 years of age, and there certainly is a sex bias that has been well documented through numerous studies. With AMR, you will get that population hit, but you'll also get things like newborn and mothers in labor and people with immunodeficiencies. And so there is a slight difference going on here between COVID-19 and essentially antimicrobial resistance, depending on, of course, where AMR is and what type of bacteria it is in. AMR will have the greater health and financial burden. Now, at the moment, that's not on anybody's radars because nobody cares about it. We're all running around trying to work out how to get out of COVID-19. The health burden of AMR will fall to low middle income countries. And that is actually entirely different to what we're seeing with COVID-19 for a great many reasons. But the burden of AMR, rather like COVID-19, will almost certainly come to high income countries. And so we're going to have to find a way to reconfigure, if you like, our economic situation and forecasts to try and deal with this, because the world will be looking at the West to try and find out how to finance all these sorts of things. So I think COVID-19 has woken up the world, not least the layman person, to how important infectious diseases are. And so I think there is hopefully new momentum to study antimicrobial resistance. And I've called it that actually AMR is like the slow tsunami, but actually COVID-19 is the sort of the rapid earthquake. Anyway, that endeth my talk. Hopefully it was vaguely interesting and enjoyable, and happy to take questions at the end of the session. Thank you.
That was great. Thanks very much, Tim. We'll move on to the first of our 3 short talks. And first we've got Evangelos from the University of Bath.
Just share my screen. Can you confirm that you can see my screen?
Yep, we can see your screen.
Is it in my presenter mode? Right. So I'm Evangelos. I'm a postdoc at the Sam Shepherd's group at the Millen Center for Evolution of the University of Bath. And I'm going to talk about multi-drug resistance in Campylobacter isolates from multiple sources. So Campylobacteriosis is caused by Campylobacter. It's estimated to be around 200,000 cases in Europe, according to the last ECDC report in 2018, and that's per year. Most people are familiar with Salmonella, Listeria, E. coli infections, much nastier, and they cause disease, you can die. But actually, Campylobacter confirmed cases are surpassing all these three infection cases combined. And when you get Campylobacter, it's usually self-limiting, so you don't go to the hospital, so it's a lot underreported. And experts estimate that there are around 9 million cases every year in Europe. So this is an AMR symposium meeting, and the CDC has included drug-resistant Campylobacter as a serious threat in their last year's report. And with a widespread resistance, it's no surprise that WHO has listed Campylobacter fluoroquinolone resistant as high priority in the recently published list, for which new antibiotics are urgently needed. Right, so Campylobacter is not, it's usually self-limiting. You'll have some vomiting, diarrhea for 3 to 5 days, but then you'll be all right. Now, when you have persistent or severe cases, you have to take antimicrobials. So ciprofloxacin is the drug of choice, with second being erythromycin. But humans do not really carry naturally Campylobacter in their gut. So the question is, where is the selection for resistance? So this is from the CDC's same report. So in 2018, we're looking at the 2 most commonly species of Campylobacter causing disease in humans, with first being Campylobacter jejuni, followed by Campylobacter coli. So just to remind that multi-drug resistant, an isolate is considered multi-drug resistant when it's resistant to at least 3 different classes of antimicrobials. And we see that in jejuni, there's a proportion of isolates being susceptible to antibiotics, a large proportion being resistant to 1 or 2 antimicrobial classes, and a small proportion being multi-drug resistant. Whereas in coli, we see a larger proportion of isolates being multi-drug resistant. And more importantly, we see that in coli, we have high levels of AMR resistant to both these antimicrobials that are used in the clinic in severe infections. So where does Campylobacter usually live? So lots of livestock animals and wildlife, they carry Campylobacter in their gut. We also see Campylobacter in the environment. So we know that Campylobacter can be host specific, so there are particular clusters of Campylobacter associated with specific animals. So the question is AMR acquired in animal hosts? So again, from the ECDC's report, we see kind of the same trends in isolates Campylobacter from animals. And then alarmingly, we see that a lot of the Campylobacter coli ones from different animals, they have high levels of resistance against multiple antibiotics. And again, we have high levels of combined resistance to the 2 antibiotics used in the clinic. So transmission dynamics of Campylobacter can be very complex. You see, we have a lot of opportunities: livestock animals, wildlife animals, companion animals, humans, and we have also the environment. So there have been outbreaks of Campylobacteriosis associated with water supply, contaminated water, and also studies that have measured the resistance of Campylobacter isolates from different sources in the environment. So what is the role of the environment in transmission or spreading of AMR? So broadly, when we talk about spread of AMR, we're talking about clonal transmission, which is when you have a specific strain, clone, but they had resistance, and then they changed from one niche to another while transferring and retaining that resistance. And then we have the gene pool transmission, where we have actual movement of genes rather than clones into different multiple genetic backgrounds, facilitated by horizontal gene transfer. So this study was published recently, so last year, in Environmental Microbiology. So Sam's group, together with Spanish collaborators. This is the EOI, if anyone is interested in reading the paper. So Diego, which is the other co-first author with me on the paper, and the Spanish collaborators, they sampled, or they took isolates from different sources, so from different livestock animals, human clinical isolates, and also from sewage. So they cultured and they saw the isolated Campylobacter jejuni and Campylobacter coli, and then they tested them in the lab to see the resistant profile against different antibiotics. So they saw the same thing, so increasing resistance or multidrug resistance in Campylobacter coli isolates, and we wanted to see if we have low diversity AMR clones, or if we have the gene pool transmission, so a lot of AMR distributed across the population structure of Campylobacter. So even though we had such a low sample size, we managed to get a good grasp of the diversity of Campylobacter jejuni, at least, but also coli. And we see that we have AMR distributed across different clones, and that C. coli has a lot of MDR. And then we wanted to see what are the genes. Do we have any genes that they are associated with resistance? So we sequenced, screened our isolates for resistant genes and non-resistant genes. Then we found different genes present. We found 18 AMR genes, and again, in C. coli, we had clusters of those genes. So we know that AMR genes, they're usually found in close proximity in the genome. We know that plasmids, one of the ways to facilitate movement of genes within bacteria. So a lot of these, what we call genomic islands, were found located on plasmids. And then we wanted to see where do these specific clusters, so we saw colocalizations of specific genes, like this one, and we saw that these were isolated from different animals, so from different sources, also in the clinic, but also in sewage, in the environment. And then we saw this particular cluster that was not only isolated from multiple sources, but also from different, from both coli and jejuni, so from different species. So Matt has talked about, and Tim has talked about the globalization and movement of animals and the movement of people. So in a previous study with Spanish collaborators in Sam's group, they looked at this ERM-B gene, which is a gene that's conferring resistance against erythromycin, which is the second drug of choice. So they saw that there's a movement of different alleles of that gene across different continents and between different sources, and also between different bacterial species. So this gene was first isolated in China in 2014. 2 years later, Diego and his team, along with Sam, has found it in Spain. So we wanted to look at the alleles in our antimicrobial resistance genes. So looking at the tetracycline, for example, there's a lot of different alleles. It's a high diverse gene, but these are the most dominant one. So we see alleles like the blue one that are shared between different sources within the same species, but we also see other alleles like the red one that are shared not only between multiple sources within one species, but also across species. And we also see that sewage is potential route has to do with the transmission. So to summarize, we have overlapping gene pools. We see allele sharing between different species, and this is all consistent with horizontal gene transfer facilitating this movement of genes. Cannot exclude, can be both clonal transmission and gene pool transmission. We cannot really explain the coincidence of rising AMR animals and humans because we don't really have source link dynamics. But these are real world data, and does so that there is sharing, and that also it suggests the potential of sampling sewage to monitor the AMR that's entering the environment and try to combat the rising resistance, not only in Campylobacter but in different isolates of different bacteria. Lots of people to thank, the group, Spanish collaborators, and other people involved in different stages of the process, and you for listening. I'm happy to take questions at the end of the Q&A session.
Great, thank you very much, Evangelos. We'll move on to our second talk, which is being given by Kelly from the University of Exeter.
Hi everyone, can you see my screen okay?
Yeah, that's all good.
Brilliant, all right, well, yeah, hi, my name's Kelly. I'm from the Centre for Sustainable Aquaculture Futures at the University of Exeter. And first of all, just to say thanks a lot for inviting me to give a talk at this session. It's a really interesting talk so far, so I hope I can live up to that with mine. Yeah, I'm going to talk about AMR in Bangladesh aquaculture, which is the work that I've been doing. And the first thing is just to go very briefly over what is aquaculture, for anybody that doesn't know. So it's the farming of aquatic organisms, which is quite a wide variety of species. It's, as you can see on the right hand side, it's the fastest growing food sector globally, and it's sort of hailed as having great potential for food security because the feed conversion ratio for fish is much lower than in other animal production sectors. So so it's a global industry, and I've had quite a few AMR researchers saying to me in the past, oh, you know, it's a bit niche aquaculture, you know, is it really that important? And I think it is niche if you, from a UK perspective, because we know about salmon farming in Scotland is quite a big industry there, but other than that we don't see that much of it in our everyday lives. But actually in Asia particularly, aquaculture is a huge industry. About 90% of global aquaculture happens in Asia, and in Bangladesh, where I've been doing my work, it really directly impacts on people's livelihoods. So around 15 million people, that's about 11% of the population, depend on this sector for their livelihoods. It contributes 2% of GDP, so it's, you know, important economic sector. And over 50%, over 60% even of the dietary protein comes from an aquatic source, and that's the whole population. And so that's aquaculture and fisheries, but aquaculture is a big part of that. So it is niche in the UK, but from a global perspective, this is really important. And in terms of specific AMR risk, and this is a sort of cartoon of a small scale aquaculture farm. They range, a lot of them are still like this, but you also get huge and massive intensive farming systems as well. So as Tim said in his talk, Asia is a big sort of area that poses great risk to AMR dissemination globally. And one big part of this is that there isn't widespread sanitation or wastewater infrastructure. So, for example, if you look at this farm, this water that's coming into this aquaculture pond is likely to be contaminated by upstream industrial pollution, hospital pollution, towns, you know, all sorts. So it's likely to have heavy antimicrobial pollution and also of chemicals, but also pathogens and resistant pathogens as well, all coming into this pond. And that means that there's often heavy burden of disease, and this is aquaculture, the industry is intensifying as well, so even higher burdens of disease each year. It seems to go up and up, and that means that people are more likely to use antibiotics to keep their animals from alive when there's an outbreak. So antibiotics are used in the animals that are often around as well, and in the humans, and feces from animals and humans in these type of ponds go into the water quite often, and the humans have direct exposure with the water. So you have this sort of melting pot of human and animal pathogens, obviously environmental microbes, and antimicrobial pollution. And of course then this all goes straight back out, untreated, back out into the local aquatic system. And in the case of shrimp, because this is a big internationally exported product, this is all often transported around the world. So you can see that this is, this does pose a significant risk to AMR dissemination. I think I've said most of this. There is a heavy burden of disease with poor access to diagnostics and advice. And Tim touched on this as well before. The antibiotics are widely available with very little regulation of the sales and usage and the quality as well. So it's a very different situation to what we have in the sort of developed world. So when I started working on this about 3 years ago, I came in from a bioinformatics perspective, and we're still doing all this work now and looking at a lot of the spread of the genes, but this is just to give you a sort of taste of what we've been finding. These are shrimp hatcheries. So these hatcheries are part of the shrimp sector. They provide the larval animals for the ponds to grow up. And in the average shrimp hatchery per year, they're using about 100 kilos of antibiotics. And that's actually only, the season is from January to August, so that's only actually about 8 months long. So it's a huge amount of antibiotics. And when we looked at the resistance genes, this is from metagenomic analyses, there's a wide range of, and these are just 4 hatcheries, a wide range of genes. And if you look at the class from the WHO classification, they are all either critically important or highly important classes of antibiotics that we found resistance to. So you can see the science, we approach it from a scientific perspective. There are genes there, and this is a risk. But and like I say, we're doing lots more analyses like this. But because I wanted to talk about the interdisciplinary side of this. If you actually look at, we found the science that says this is posing a risk. So what can we actually do about it? And I think to think about that, you have to look at the bigger picture. So, farms and hatcheries, they are the end users of antibiotics, but you have to look at the rest of it. So obviously farms are using larvae from the hatcheries. Both of these are using antibiotics supplied by farm supply shops. And just like farmers and hatchery owners, farm supply shops, many of them are just trying to earn a living and to put food on their families' plates. They're not highly educated often. They're not trained. They are often influenced heavily by the pharmaceutical company agents and the feed manufacturers who supply all of this. The government are supposed to regulate, well, they do have regulations, they're just totally under-resourced, so it's very difficult to implement them and provide training. So it's a very complicated situation, and I think a lot of the pressure often goes on farmers and the end users when actually they often don't have much choice, especially when the environment is heavily polluted and they're just trying to make a living. So to talk about the interdisciplinary projects that we've been working on, I was just going to talk about 2 very quickly. One is really looking at biosecurity, because actually if you improve biosecurity, you're obviously reducing the need for antibiotics and you're reducing the spread of disease. So biosecurity is often very poor, so So this is an obvious place to really make some gain in terms of disease and AMR. And the second project is looking at the antibiotic value chain, because if you really want to improve the sales and the regulation, you really need to understand what's going on in the first place. So the first project is looking at the biosecurity. Again, this is looking at hatcheries, and with our in-country partners, we are collecting biological and physicochemical data to really understand whether the biosecurity risks are in individual hatcheries, to really understand how people could improve this and reduce the disease. And we're also collecting socioeconomic data to understand management practices and the economic implications of improving biosecurity, because obviously that is ultimately what is going to drive changes, is if you can show an economic benefit. We're producing digital training materials, so a range of videos that are, a lot of the training materials at the moment are in English. They're very heavily text-based and very technical, which obviously isn't very accessible to the majority of people working in hatcheries and on farms. So this is just where they were on-site shooting footage for the videos recently. And then we're also producing a self-assessment tool, so that we won't be in English, but just to give you an idea, people can self-assess their own biosecurity and see how they could improve it. And these sort of things don't actually exist, and certainly not specific to their industry and not in their local language in an accessible format. And then very quickly, because I know I'm pretty much out of time, the second project is looking at the pharmaceutical value chain. So we're working with in-country partners, we're interviewing shop owners, and we're having focus group discussions with government officials and pharmaceutical agents, farmers, all different stakeholders in the value chain, to really understand how the whole thing works together, how all the stakeholders interact. And this is very preliminary data, but like I say, we're collecting all sorts of data, but this is a really nice way of seeing the relationships between the stakeholders. So the thickness of the line shows the sort of frequency of the interactions, and you can see the relationships between all the different stakeholders, and then the transfer of money, information and goods. And just with this and other data, we can really get a really full understanding of what's going on and try and design interventions. That's the next step to address this issue. So if there's only two messages, first one is aquaculture isn't niche. It poses a significant risk to AMR, and an interdisciplinary approach is key. And just to very quickly plug our Exeter network, which I've been helping run for the last year, and we hope we can integrate well in the new GW4 AMR network, whatever you call it. Okay, and thank you very much for listening.
Thanks very much, Kelly. We'll move on to our third and final talk for this session, which is from Jordan from the University of Bristol.
Can you guys see my screen okay?
Yeah, I can see that.
Yeah. Just do slideshow view. There we go. Okay, yeah, so I'm Jordan Seeley. I am doing a PhD at the University of Bristol, and I'm going into my third year now. My background is in zoology and infectious diseases, and this kind of combines the two together. So I'm not going to go over too much of what Matthew has already said, spoken about today with the OSTAR project. But basically, this PhD has arose because of the discovery of the multi-drug resistant plasmid, that is PMU32, which, just to quickly recap why this plasmid's interesting and important to research, it carries resistant genes to streptomycins, aminoglycosides, tetracyclines, but also to the critically important third generation cephalosporins. And so infections to this plasmid would be difficult to treat, whether that be in animals or in humans. The gene responsible for this cefotaxime resistance, just to go over, is CTX32, which is uncommon. The common variants of this are usually 14 and 15. Yet this plasmid was responsible for most cefotaxime resistance on these farms and accounted for nearly half of the farms within the study. These farms were also within a particular region of mid-Somerset, so they were all found within a 20-mile radius of each other. And considering the plasmid is very large, you would assume that it was costly to the organism to carry. But due to its success on these farms and being found in multiple sequence types, it's clear that it is benefiting the bacteria in some way. So something is obviously owing to its success, and my research aims to discover the factors selecting and maintaining this plasmid on these farms, and also to determine whether it's present in the surrounding environment and local community. So to address whether the plasmid is present in the local community, I chose to look at gut commensal E. coli from dogs within the region. The rationale behind using dogs as the example for the local community is, firstly, since they live with humans and have regular contact with their owners, this allows for the sharing of bacteria, including those that are resistant. And secondly, they have greater contact with their environment than their owners. So in the environments that they're walked in, more so than the owners themselves. And this increases the opportunity of picking up bacteria within these environments, and particularly if they're walked around the farms or through farmers' fields or the footpaths, this can allow for the opportunity of picking up this plasmid. So my first and foremost aim is to select for E. coli isolates that are resistant to cefotaxime, as this will select for the for isolates with the PMU32 plasmid. And then the wider aims of this dog study will look at the other mechanisms of cefotaxime resistance, resistance to ciprofloxacin for fluoroquinolone resistance, as another measure to have a look at humans, animals and farm transmission. And lastly, nitrofurantoin. Now, this antibiotic is only used for human UTIs, so we wouldn't expect to see it in dogs. However, if it is present, we would then it would then indicate that there is sharing of bacteria between humans and pets. I then carried out the same study, but with city dogs, to add to the data of resistant genes and sequence types between dogs, and this will be compared with that of rural dogs. And also these data sets will then be compared with already collected data for human bloodstream and UTI infection, so the E. coli that's been found in them, but from the same region as well. So on to the sample collecting side of the dog study. First, both human and animal ethics were approved, and then basically what I would do is pick a location within a 10-mile radius from the central point of the region where this plasmid was present on the farms, to increase my likelihood of finding the plasmid within the local dog population. And also I'd set up in areas that would most likely have dogs being walked. So this included parks, footpaths, commons, villages, and also a local dog show. And prior to going out, I'd set up collection packs, which would include the participant information sheet, a questionnaire, which would lead to perhaps some risk factors for carrying resistant bacteria, consent forms, because consent needs to be gained in order to take a sample from the dog, which I probably should mention. If you haven't already guessed, it's a fecal sample to have a look at their commensal gut E. coli. And also then a biohazard bag to safely transport it to the University of Bristol. So to participate, owners were required to complete the questionnaire, consent forms, and provide the fecal sample from their dog. And for those who wanted to get involved but their dog couldn't provide the sample at the time, we handed out postal packs, and then they were sent to Bristol to be analysed in the lab. So the risk factor survey included questions to determine risk factors for carrying resistant bacteria. And this included the type of food that they eat, the environments they're walked in, and whether they go into any water sources, and if they've been unwell in the last 6 months and therefore had antibiotics. The fecal samples were then taken back to the lab for processing, and I plated these onto selective antibiotic plates, being cefotaxime, ciprofloxacin and nitrofurantoin. And overall, on the dog study, I collected 301 rural dogs, samples from rural dogs, and 298 from city dogs. In the rural dogs, 11.3% were positive for resistant E. coli. Of this, 59% showed cefotaxime resistance and 65% ciprofloxacin resistance. No nitrofurantoin isolates were identified. And in the city dogs, 17% were positive for resistant E. coli. Of this, 62% was cefotaxime and 70% ciprofloxacin. And I did discover nitrofurantoin resistant isolates in 2 dogs. So this is the first time that it's been identified in these dogs, and until I've analysed the whole genome sequencing data, I don't know about the sequence type or the resistance genes they're carrying, but once we analyse that and we compare it with the human E. coli isolates from human UTIs from the same region, we can see if there's any similarities with there or any sharing of these isolates. A maximum of 3 isolates for each selected resistance per dog was then analysed for, during multiplex PCR. There were 4 of these. First to determine the PMU32 plasmid, other beta-lactamase genes, the CTXM group, and also plasmid-mediated fluoroquinolone resistance. And the results from these, so I've found 2 isolates positive for PMU32. So the plasmid, which was found on farms during the OSTAR study, a dog within the area of these farms has come up positive for the plasmid. Whole genome sequencing will confirm whether it is the same sequence type and same type of resistance, but it's looking pretty sure that it is. And we don't know whether this is maybe a farm-like E. coli that's just gotten to the dog and is passing through and I've managed to grab it at that time, or whether it's actually transmitted into its commensal gut E. coli. So we'll soon find that out. But this potentially confirms our hypothesis of the plasmid circulating within the local community and could be an indicator for circulation amongst the human population. For the rural and city dogs, there was different carriage of resistance genes at varying levels. For example, there was 4 times TET-B, 3 times more CMY, 5 times TM in isolates from city dogs than that in rural dogs. Genes DHA, CTXM, group 25, OQXAB and AAC was identified in city dogs but wasn't found in rural dogs. But contrastingly, CTXM group 9 was seen 7 times more in isolates from rural dogs than city dogs. However, this, whether there is any significance in this is yet to be determined by statistical analysis, but just eyeballing the results, that's what we've seen. And for, the quinolone PCR, not a lot of genes showed up in that. So it's for plasmid-mediated genes, so this is predominantly chromosomal resistance. So just to summarise the dog study, PMU32 has been found in a dog in the area of the farms, and nitrofurantoin resistant isolates have been found in dogs in the city. So the rest of the work that's then going to happen with this, I'm going to be running risk factor analysis for resistance in dogs using the questionnaire that was completed by the owners, statistical analysis between rural and city dogs to see how these compare, and the whole genome sequencing analysis of resistance genes and sequence types for all of the dog isolates. And then these will be phylogenetically mapped and compared between human, dog and farm isolates from mid-Somerset region, and also human, dog and zoo isolates from the city of Bristol. which I should mention, we've collaborated with Bristol Zoo to have a little look at the resistance genes that are carried by the animals, the captive animals in their zoo, and to see whether, with it being a city zoo and they're in close contact with humans, whether they share any resistance genes and sequence types of E. coli. Thank you very much, and I look forward to any questions.
That was great, thank you, Jordan. So we're actually running a bit late. The panel discussion was meant to finish at this time, so I think what we'll do is I'll ask a couple of questions to the panelists if they're happy to stay, and then we'll finish and just maybe have 10 minutes for the break instead of the, I think we had 25 scheduled. I was wondering, would all of the panelists who are still here be able to turn on their videos? Unfortunately, Tim had to leave, but we've still got 4 or 5 people. And I will read a couple of the top questions from the Q&A. We have, just to let everyone know, we have had a couple of questions that have been submitted and answered already, so you should be able to view those. But I'll just start from the top. And the most voted question is, I think it's to Matthew. Do you think the changing farming practices in the UK or globally, i.e. increase in organic and free-range farming versus intensive farming, will impact on presence and transmission of AMR, as you mentioned, location of pasture versus housing difference?
Well, that's a $1 million question, I think. We did include some organic farms within our study. There was no suggestion that they had less resistance than not. In fact, there was no suggestion that they had actually much less usage of antimicrobials than some that weren't organic. So I think that in itself is not a recipe for solving the problem. Intensive, well, that's an interesting question. Of course, some intensive systems actually use less antibiotic because it allows for closer monitoring of the animals. I run projects in Argentina and I run projects in Thailand, and of course it's an entirely different type of system there. So in Argentina we're talking about much more intensive systems, feedlot systems for finishing. In Argentina much more extensive, in Thailand much more extensive but small scale in the way that Kelly described for aquaculture. So there's not like a one size fits all, I would say, and I think you've got to factor in usage, of course, because you've got to drive resistance, usage of antimicrobials, but not just that. Heavy metals and things like disinfectants can also potentially select resistance. I think one of the biggest issues is likely to be transmission of bacteria, and that's around hygiene. Again, I think Kelly picked up on this idea that bacterial transmission from one farm to another, from within the farm, from crops of animals, that are in and out systems. Those are some of the areas that I think we probably can learn most around. And could actually have a big impact, and obviously the same goes for humans as well, frankly. Usage is important, but actually hygiene and clean water and all of those things are perhaps more important in certain parts of the world. So that's a long-winded way of saying I don't really know, but that's the answer I'm giving. I don't know if anybody else wants to chip in and comment.
Can I add to that? Yeah, I agree. I think, and I saw a talk recently from a group in Sweden, I think it was, and they looked at the difference in Thailand between intensive and less intensive farming, and they found that there's more antimicrobial resistance genes on farms that are more intensive, because I think the idea is they're using more antibiotics, but actually in terms of transmission to humans, it's less, because there's less human exposure. So it then raises the question of what's greater risk? Is it the use of antibiotics, or is it the fact that the human pathogens are there as well? So I think that's really interesting to look into more.
I actually have a question from one of the panelists to Kelly, which won't have appeared in the box because it's from someone in the group here, but I'll ask it because I think it's really interesting. How much of a role would consumer label certification schemes like MSC or ASC play in tackling AMR and aquaculture?
Yeah, again, that's a really interesting question, because I think, so in Bangladesh, the shrimp industry, shrimp are exported, and so the farmers of shrimp are under more pressure to reduce antibiotics from the government, because we have checks on when things come, the imports coming into the EU and America, we have checks on for antibiotic residues. So the export sort of checks actually drive improved behaviour in many ways. I mean, there's ways that farmers get around this because they add loads of antibiotics and then they don't add it 2 weeks before harvest. But anyway, there's pressure there. And again, certification schemes could therefore be useful for export in the same way. However, most aquaculture, so 90% of aquaculture in Bangladesh actually is for the domestic market, or what they call South-South trading, which means it goes to other low-income countries where there aren't any sort of, these drivers aren't in place. So, and certification schemes, really, you know, you're talking about people who they're buying food as cheap as they can because they are poor, and so if you have to pay a bit more for certification schemes, I'm not sure how much that really will impact on things. But I think it could, and that's one thing we're looking into, but a lot less than it would in our sort of society.
Looking through all the questions, we've got a lot for Matthew. I'll do the second one down the list for Matthew. Do you have any idea why you see this increased prevalence of resistance in the warmer months when it's not due to more E. coli being around? Could it have something to do with the activity of competing microorganisms that might be exposing your E. coli natural antimicrobials?
The answer is we don't know that. That's an interesting suggestion, whoever put that in. Another possibility is that much of this resistance is plasmid mediated, and plasmid transmission may be dependent upon temperature, and so the rates of transmission from one organism to another may be lower at lower temperatures. And another possibility is fitness cost. So antibiotic resistance imposes potentially a fitness cost, particularly plasmid mediated resistance, and it may be that that fitness cost is greater at low temperatures than at the temperatures we usually measure it at, which is in a lab at 37 degrees. So these are kind of working hypotheses that we would be very interested in testing actually by doing experiments, and have bought a refrigerated incubator for the purpose.
Great, thank you. Okay, I'm going to end with one last question that I think is kind of addressed to everyone. And it is, given the low frequency of ongoing transmission between humans and animals, shown by Matthew, the zoonotic origin of many pathogens in AMR, discussed by Tim, the commonality of gene pools, shown by Evangelos, and the complex social dimension, discussed by Kelly, is it time to explicitly define what the One Health problems actually is or are? Quite a lot to answer in that question, but I'll open it to everyone.
Well, I'll say, to avoid silence, that is it? Yes. I mean, the question sits at the heart of the issue, which is that AMR is not one thing, and there are lots of different bug-drug combinations, and in terms of resistance, in terms of transmission, in terms of importance, in terms of zoonosis, and so on, in terms of where you are in the world, frankly. So that's the first point. But the second point is I personally don't think of One Health as being a series of questions, but more a framework by which you can do research. And I think that what that framework is clearly under debate sometimes, but basically it's a way of thinking of the problem, but you may have to ask very specific questions according to the system that you're working on, and not actually extrapolate too much from that. But that's as good as I can give you on that one.
Just to build on that, I think it's also a fact of a lot of different disciplines working on the matter. So from getting the hospital, giving the right samples, getting the vets, getting the right samples, collecting the samples, designing the study from the beginning, from culturing the right isolates. So for Campylobacter, for example, which one to pick? You're going to take a scoop, you're going to do a metagenomic, how are you going to approach the problem, to further on sequencing and looking at the AMR. So it's a lot of different people working together, and that can cause a mess.
Yeah, I just add to that I think actually AMR it's more than it's just one problem. It's actually should be a sustainable development goal, and I know they're talking about this already, but unless we address sustainable development, we're not going to address AMR. And so it either needs to be a theme that goes across the sustainable development goals, or it needs to be a goal in itself. So I think Ed's right. I think it was Ed's question. Yeah, it's everything basically.
Great. Thank you everyone for your contributions. I think we're going to finish then. There are some unanswered questions, which if the panelists have time, they're more than welcome to answer themselves by answering them live, or if they're happy to, we can put them on the discussion forums on our website to continue the discussions then. So I was going to finish the session by just saying thank you to everyone, and thank you to everyone in the audience for their questions. We're going to have a short break until 3:20, and then we'll start our second session, which will be hosted by someone else in the organising committee. So if everyone would like to go away and have a cup of tea or do something else, or look at our website for, we've got about 10 minutes, then there'll be other panelists coming in that will be setting up and testing their video and audio for the next session. So if you hear something, we are testing people, but we won't officially start until 3:20. So thanks again, everyone, and we'll see you when we come back.